Good evening, everyone. Am I audible to you all? Please confirm in the chat box. Yes. Hello, Bhumi. Hello, Ayush. Welcome to you all. Yes, who else do we have here? Yeah, very good evening, Shabini. All right, Ashish. Very good. Very good. Thank you for your responses, everyone. Ansh, I'm super excited to see you all here. And I welcome you all to today's webinar, Bioinformatics Career Ladder, from internship to industry level jobs. Let me begin by asking you something very simple. Have you ever felt lost, wondering what comes next after my bioinformatics course or internship? Or maybe you have thought, how do I even get my first break in this field? What is the next step? What do I do next? Yes, I'm sure if you have thought so, then you're not alone. I am sure many people have thought like this, right? Yes. Good evening, all of you. Renu, Nidhi, Kritika, Geeta, I welcome you all. And it's really good to see you all here. Right. So you're not alone because thousands of students and researchers they ask this question every day. And that's exactly what we are going to clear up in this session. Yes, that's right, Shabani. So what is this webinar all about? Today's session is all about your career journey in bioinformatics. Be it your very first internship that you apply for, or growing into a scientist or a data analyst, or even if you want to climb up the research ladder in a big company. So, we'll talk about how to make the right moves early in your career, what skills matter the most in the industry, and how people like you have grown from beginners to experts. So, why is this topic so important? Because bioinformatics is not just about writing code or analyzing DNA, it's about solving real problems like designing better medicines, understanding diseases faster, or even tracking new pathogens. So, here's the thing science may be exciting, but the career path can be confusing at times. So that's why today we are breaking it down into very three simple steps. How to start your career, that is internships, training, first roles, basically the internship and entry points, how to launch your career. Second is how to accelerate your growth in the mid career strategy, how to grow when you are some midway, what strong skills you have to build, or how can you switch the roles, and finally how to lead as a senior expert or manager. So we will be looking at the executive pathways, how to drive strategy and innovation, how you can lead at that level. So whether you are still in college, just starting out, or already working, this session is definitely going to benefit you. So, what to expect today? We have three amazing speakers who will each take you through each of the steps of this bioinformatics career ladder. Okay, so they will share real tips from their experience, their industry insights, and things that they have learned through years and years of experience. So get your notebook ready, keep your questions coming in the live chat. We will be taking the questions at the end. Stay with us till the end for this part. Okay, so please help me welcome our speakers for the day today. Our first speaker for the day is Ms. Snigdha Tiwari. So, ma'am is a senior research fellow at IIT Roorkee with 5 plus years in computational biology, bioinformatics, and telemedicine. Specializes in protein interactions, molecular docking, and clinical data management. Ms. Tiwari has led the development of the Utsarjan app for pediatric nephrotic syndrome research with AIMS Delhi. Currently, she is pursuing a PhD in computational biology. She is also an academic support specialist in Biotecnika's Bio IT and Clinical Research Department, bringing interdisciplinary expertise in multiple subjects like chemistry, bioinformatics, and biotechnology healthcare innovation. Welcome, ma'am. Our next speaker for the day is Dr. Neeraj Kumar. Dr. Neeraj Kumar is a computational biologist who uses artificial intelligence to speed up drug discovery. He earned his PhD in bioinformatics from CSIR, IHBT, and SIR, then completed his postdoc research at Penn State University, where he built algorithms for antibody analysis. Dr. Kumar is skilled in tools like TensorFlow, PyTorch, RDKit, and GROMATS. He has published in leading journals such as the Journal of Cheminformatics and Medicinal Research Reviews. He has also cleared competitive exams like UGC NET, GPAT, NIPER JEE, and GATE as well. Welcome, sir. Our third speaker for the day is Dr. Girisha Verma. Dr. Verma is a senior business analyst and product owner at Daiichi Sankyo, where she leads bioinformatics strategy and data integration efforts across global translational research initiatives. With over 6 years of experience spanning biotechnology, data analytics, and pharmaceutical informatics, she specializes in bridging biological hypothesis with computational execution. Ma'am holds a PhD in biotechnology and is certified in business analytics and drug discovery and development from Harvard. Dr. Verma plays a key role in managing large scale scientific databases and AI driven pathology image platforms. Her cross functional leadership includes designing digital workflows, driving regulatory readiness, and translating multi-omics data into actionable insights for clinical and R&D teams. We are honored to have you here, ma'am. Welcome to the session, and we look forward to your session as well. So, what are we waiting for? I think we are good to go, and without much further delay, let's hear it from the experts. So, I hand over the controls to our first speaker, Snigdha ma'am. Over to you.
Thank you so much, ma'am. Uh, Hi, everyone. Good evening. I'll be talking about, the skill mapping, the networking, how to, how to start and land up to the career in academics and industry. So quickly, let me share my screen to you. Okay. So, guys, today we'll be talking about launching your career. How the internships and entry points, in different levels. The academias, the contract roles, the industry. How do you do the internships, and what will be the entry points? What will be the network and skill mapping that you need? What is the need of an hour? We'll talk about, and definitely, how transitioning your career, from academia to industry, in especially in bio IT roles, in healthcare diagnostics, bioinformatics, computational biology, and all of that. So starting with the top entry routes into the industry, what can be, your top picks, what can be, the icebreaker routes of your, of your future. So there are three common entry points, in especially in the bio IT and bioinformatics roles. That is internships. Through internships you can definitely get into, the academia, many of the project roles, or the industries. So these are the easiest and most direct ways to the real world exposure. They can be with any of the startups or academic industry collaborations, or the biopharma companies. Of course, I mean, there are a lot of collaborations that is happening, with the good academic roles, with with the industry and everything. So the second way that you can get into is through graduate and leadership development programs. So offering by larger organizations, these are some of the programs, that is offered by the organization, some big organizations. The names are Roche, Novartis, and Illumina. So these are structured and rotational, often leads to the full-time jobs. Generally, people underestimates the, the contract or the the temporary roles. But that is actually the big, deal with you landing on the permanent positions, of course, because they are, these may be through staffing agencies. The contract and the temporary roles may be through the staffing agencies, or they can be project based roles initially, but through short term, though they are short term initially, but they offer the valuable experience and networking opportunities, and often they turn into the permanent roles. So never underestimate the contract temporal roles that that may come to your that may come to your way. Talking about the skill mapping, how skill mapping actually helps you, and what are the what are the topics, and what are the area of academic tasks, or the skills that that you should keep in mind, that is trending, that the roles that are trending, the skill mapping that that can help you translating your academic work. Many of you may be doing the academic work, in the collaboration with a few labs or the academia. So how can you actually transition from academia to the industry? So that we will also see. So if say at all you're doing your thesis in say for example in cancer genomics, then you can definitely land up into the translational bioinformatics or biomarker discovery roles in the industry. If at all you are doing the RNA sequencing using bioconductor or Python, if someone is doing the the analysis in in a project or in the academia setup, then the one can land into the roles like NGS pipeline analysis and automations, and all that industry roles. Again, if someone is doing the development of the tools for sequence alignment, or developing the algorithms and tools, then one can land up to workflows, optimizations, computational efficiency, and all of that in the industry roles. So watching all these academic tasks that that help you map the industry skills, it is very important to to start somewhere, to start with maybe any of the contractual roles, or the projects, or in the collaborations, something that will definitely help you. Say for example, if in the academia task someone is doing the group projects or hackathons, that will definitely help you in the industry with the agile teamwork, spirit based deliveries. Someone there are a lot of students, there may be a lot of people listening to me are interested in literature reviews and reports. They are experts in writing and reviews and reports and publications, so they can definitely land up to regulatory documentation rules, SOP writing, scientific writers, and all of those industry roles. Again, mentoring juniors, TA. If someone is actually very good in TAs, and mentoring your juniors and all of that, or delivering the lectures, then then you can land up to communication, cross functional collaborations in the industry skills. All this have given you a sneak peek to how your academic task can can help you land up to the industrial skills and the industrial setup. So top skill sets to focus on, there are few skill sets that I've tried to uh, try to concise here on my screen for you, that what are the top skill sets that you should definitely focus on in the current scenarios, that will that will definitely help you landing to all of the roles that that we've talked about in the previous screens. So here, some of the skill areas that I want to talk about is data wrangling and analysis. Data wrangling and analysis, the key tools that you should focus is Python. Some of the R like dplyr and tidyverse setups and libraries. Then there are a lot of learning resources as well, like R, Python, Kaggle courses. There are a lot of data camps that run. Uh, uh, the skill areas could be the bioinformatics pipeline. The key tools that help you set up the bioinformatics pipeline will be Nextflow, Galaxy, shell scripting, and all of that. A few of the learning sources I've concised for you is Galaxy trainings, NF core tutorials. Definitely, you can check these. Go to their pages and check what are they providing to you. NGS and genomics, if someone is interested. RNA-seq analysis, DNA sequences, alignments using star, Bouty, BWA, variant calling, all of that. Then some of the resources are Coursera, genomic data sciences. Uh, more of the skill areas, talking about more about the skill areas, version control, it is very important. The key role and the concepts, git, and how to pull the request, how to make your own GitHub library and your own GitHub documentation and everything. So that is so important. Some of the few of the learning resources for it is here on the screen: GitHub Docs, learn Git branching, and all of that. That will definitely help you when you land with the interviews, or or showcase your skills when you want to land up any of the industrial roles or collaborative roles. This GitHub, developing a GitHub is a must nowadays. So I will definitely encourage all of you to to definitely look onto the version control things. Then the databases and querying, you can just go ahead with the MongoDB, SQL, non-SQL databases. Clinical research is actually using a lot of it these days, because as the clinical research is going to the to the big data analysis, it is so important, in the healthcare diagnostics, and all of those health tech, and those roles, those industry to land up. um, More about learning resources, if I talk about NCBI utilities, SQL tutorials, all of that, you can go ahead, look onto the pages and see what are they providing, what you can draw from there. Then visualizing and reporting, this is also a very important, these are very important key tools if you want to maintain or draw some algorithm. You have to design the biomarkers algorithm, or anything like that. Matplotlib, Seaborn, ggplot, all of that is the basics and so important. You can for the resources, you can go to the Python plot galaxy, you can look into the R cookbook, all of that. Then soft skills that will help you um, with the industry roles, with academic roles, is presentation, teamwork, and problem solving. That approach is definitely very well, uh, very well. You should be knowing that, of course. Then what? So how can you actually do that? How can you actually do the teamwork and problem solving thing? So LinkedIn learning is can be one of the path. Free with some institutions. There are LinkedIn learnings. That is there. You can build your LinkedIn profiles, you can network there, and all of that. Now coming to building your application toolkit. How do you build your application toolkit for landing up for landing up with your first say a job, or or a collaborative projects, or all of that. So resume building, if you talk about the resume building, you should tailor it for each role. You should tailor your resume for each role you are applying for. You want to do whatever you want to do, you should definitely tailor it like that. Watching Use industry keywords, term like data curation, pipeline development, then GXP compliance, if relevant. You have to definitely add all these industry keywords to your resume. Like you've developed a pipeline, or the data curation thing. And you should also I would definitely encourage you to show the outcomes in your resume, in a little snapshots. Say for example, uh, you did you've achieved reducing the processing time by 30%. And maybe you have developed an algorithm and you have reduced processing time by 30%. So all of that, you can include as a snapshot in your resume. Building your resume, LinkedIn profiles. And I've talked about the GitHub, talking about the tools like GitHub and GitLab is so important. And that will definitely land up uh, uh, encouraging the interviewer to to listen to you, and to trust your skills, and to trust your projects, what you did in a project. So highly recommend developing your GitHub library. So a must have, if you're in bio IT, upload your scripts there, upload your scripts from a thesis, or or any of the projects that you did. Even simple data cleaning notebooks, even if you annotated, or you applied any of the pre-processing or cleaning the notebook, then also you should pull it in your GitHub, and show to the interviewer that you are doing all of that, and you're experimenting with the data sets, and you can talk about it, annotating them, and show the show the main readme files also. Talking about the LinkedIn, the optimize, I've talked about it. Optimize your about section in the LinkedIn. That will definitely help you. Use your profile as a storytelling story. The profile should be crisp, and the profile should be attractive, as in you should be uh, writing, and shortly about yourself, and what you do, so that so that people just arrive on your LinkedIn profile and know what you're interested in, what you're doing actually. So use action verbs and achievements in your LinkedIn profile. um, Also suggest to network before you need a job. If you apply a job, definitely the golden rule is network before you need a job. So networking is so important, to stay connected with LinkedIn. And networking is so important. That definitely helps you. So start connecting with the professionals, and attend events like, you can also attend a lot of conferences and events. Some of you have put in concise for you, and I'm showing here on the screen few of them, like like ISMB. They are perfect for computational biology network. So you can join ISMB, you can join the BioIT World, all of that, for great, for applied data sciences and AI in health. If someone wants to join bioinformatics, health tech, AI in health, then definitely network and go ahead and join all these uh, all these hubs, and the collab, do the networking and collaboration with them definitely. Then even webinars and LinkedIn live events count. What are the webinars you're doing, how much you're listening to the experts, and LinkedIn live events and everything. You can just go ahead and engage with them. You can ask questions, comment on post, and thoughtful interactions, and that's how you get the referral. Maybe you can ask for referral. If you do well, you never know, you can land up a good job, or or a project, or things like that. Creating an online presence, that your portfolio building is so important. Once you develop your LinkedIn, and you're good to go with your GitHub pulls and repositories, you also have to do your portfolio building, and that portfolio building um, uh, you know uh, comes with all this. Your GitHub, your Medium and Substack, your Kaggle or bioinformatics challenges that you did. A lot of Kaggle and bioinformatics challenges goes on. I highly encourage you to participate, build your credibility, pull in the GitHub and show it to the interviewer. So centralize your projects, CV, where blogs and contact info. Use platforms like LinkedIn and GitHub. Few of the conferences, networking and visibility, the key conferences, these all may be some of the good key conferences that you must go ahead and check them out. ISMB, BioIT World, Recomp, ASHG, EMBO, CDM. All these are the good ones. You can definitely go ahead, check them. How can you participate, how can you network in all these conferences. Highly recommend you to do all that. A networking tip is, don't ask for a job as okay. So this is just a networking tip that I've just dropped for you, that ask for the advice. Definitely keep asking for the advice on LinkedIn while networking. That really helps. Leverage Slack, LinkedIn, ResearchGate, and Twitter. Always try to upload your LinkedIn profiles, your Slack, your ResearchGate profiles, and your Twitter accounts, or X, or whatever you use. So that all helps, networking and being active on all those social media. Of course, networking, once you have all your work done, all your skill mapped, that is you should have a stronger base. That is very important, like your GitHub repos, and your projects and everything. So what are the academic settings, and how again how do you bridge the gap with your academic settings to the industry settings? So long timelines for projects. So academic settings definitely have will give you a long timelines for projects. You may have a picks for you have a picks for 2 or 3 projects, and you have a good timeline for it, to build end to end academic settings for it. On the other hand, industry settings will be fast paced, because of course it is a delivery driven timelines it will have. It's an industry. It focuses on business values and ROI. while on the other hand, academic settings maybe, may give you the freedom to explore ideas. The research and your projects, your all of that will give you freedom to explore your ideas, bringing up your ideas with the long term timeline goals and projects that you picked up, and and finish it all. Then emphasis on novelty. Definitely academics emphasize more on what is novel, what you did is novelty, because research comes with a novelty, of course. But on the other hand, the industry emphasize on reproducibility. What are you giving them, how to scale, actually, the scalability. So fast paced industry settings, you can explore the fast paced industry settings once you build your solid academic settings. Academics are supervisor driven. It is cross functional teams and limited tool standardization is there in academia. In the industry, there are SOPs and pipelines, that you have to follow. So how so I would like to definitely talk about this. As I've talked about how you can bring up your skills of academic to the industry. I would like to talk about how Biotecnika helps you transition from academia to industry. So Biotechnica definitely skill conversion training, do a lot of skill conversion trainings, real world case studies and projects that Biotechnica upholds. A very good mentorship from industry professionals, a professional development toolkit. They provide the professional development toolkit to you, a mentor at each level, end to end level. That industry networking opportunities always there. Post internship support and placement guidance is also available. So these are some of the points that how Biotechnica definitely helps you transition your career. The real job titles, I've put some of the real job titles for you. What domains you want to achieve, what domain you want to land up, and what are the entry level job titles that you can look for. So you can look on the screen that there are different domains like bioinformatics, genomics, AIML, data science, and clinical research. So based on that, what are the entry level jobs that you can look for. I have flashing on my screen the entry level jobs. What are the what can be the what can be the titles. You can the job titles and everything that you can explore the analyst and researcher, variant analyst, and based on the biostats and cross functional domains also. There are a lot of entry level jobs that I'm putting for you on the screens, so you can definitely go look for it. So once you look for the entry level jobs in the industry, based on your interest, how do you navigate your first interview? So talking quickly about navigating the first interviews, common questions that the interviewer may ask and uh, expect from you definitely is, describe your thesis, tools, stack, pipelines that you used during your academics, the projects or academia. Interview preparation should go on like this. You should follow staff format, then then mock interviews, portfolio walkthrough. You should give a good portfolio walkthrough, what you did, like like your GitHub repositories and all of that. Then soft skill matters, for sure. How you communicate, how what are your curiosities, all that matters in your in the interviews. uh, Final takeaway tips that I want to give all of you listening to me: start small. Always start small. You can focus on the internships, short term roles that I've talked about, the contractual roles, the projects, the collaboratory projects. Translate your academic story into business values. How do you translate that? I've talked about transitioning from academia to industry, that you should be picking up very wisely. Build your GitHub, that I highly encourage, and your online presence early. Customize your resume for each role. Then networking consistently and strategically helps you a lot. So this was the final takeaway. What will be your action plan? The final takeaways and the action plan, talking about: start before you finish school. As soon as possible you should start. That helps you build small visible projects. Start developing small and visible projects. Start getting the insights for the projects you build. Then join bioinformatic communities. I've dropped all the bioinformatics communities for you on the screens. Then then keep learning. You don't need a new degree, just the relevant skills. Definitely industry and academia is actually searching for good relevant skills, and that matters. That you can showcase with your GitHub repositories, your LinkedIn profiles, your resume. So that matters. uh, so with that, thank you so much, guys, for giving me time, giving us. And I would like the next speaker. to take over. Request.
Yeah. Thank you so much, Ms. Snigdha, for those practical tips on how to land and, in fact, excel your first, be successful in your first bioinformatics internships. I'm very sure that many students wouldn't have known about the intricate details that you have shared, what all exact internships that they can look for. And obviously, Biotecnika would be a one stop place if you want to gain knowledge in this field. So, thank you once again, Ms. Snigdha. We will move on with this to our next session. Let's keep the momentum going. And now I would like to hand over the controls to Dr. Neeraj Kumar. Over to you, sir.
Hello, everyone. Good evening. So, I am Dr. Neeraj, and I think Dr. Urmi has already introduced me to you guys. So, today I am here to talk about. I am just proceeding with the what Miss Snigdha has already discussed. So, let me just share my screen with you. Just wait for a second. Can you please tell me that my screen is visible or not? Could you please?
Yes, sir, it's visible.
Okay, so let's get started. Yes, so today I am here to talk about accelerating growth, mid-career strategy, and why we need mid-career strategy by skill stacking, promotions, and lateral moves to supercharge your career. So I'm Dr. Neeraj. I already told you. So let me just take up the pen. Yes. So why mid-career strategies matter? So your first job is just the beginning. The real game starts now. So early roles helps you build foundations. They are not the terminal point. They are just beginnings. So the beginning is not your peak. You need time, you need experience, as well as you need more skills to be on your peak of your job. So a proactive strategy is crucial to staying relevant and fulfilled in your career. So your growth must evolve with your field, right? Avoid stagnation. So by mid-career, our routines and our daily habits can replace our ambitions. You cannot have new skills and opportunities without intentional planning and professional risks. So you must not just stop at one place and just keep stucking there. You need to move out of your comfort space sometimes. So skills gained early may expire with tech shift. So uh, there are various industries, mainly industry which we are working in, which is science, technology, as well as biotechnology, right? It These industries are moving at very fast pace. What worked 5 years ago may not be working longer in next 5 years. Okay, so the skills you already have while joining your job may not be fit for your job at present. So you need to evolve in your field with your skills as well as with your mindset, right? So the continuous learning is a main option to be on track, right? So companies always value uh, high skilled employees, right? So employers not just look for experience, they reward the experience as well as skills. So I have uh, I have a few skills in my mind which would be rewarded as well as acknowledged by the employer. Like if you have a strategic thinking, right? If you can strategize a specific role or a specific plan for a specific mode of action, then you might be the favorite fit for your employer, right? And second, if you have some problem solving skills at larger scale, if you have a complicated for say if you have a complicated problem at your firm where you are working right now, if you are working in your industry in a biotech lab or in a research lab. So uh, if you have to work out with a complicated or a complex experiment, then you must have problem solving skills that may catch employers eyes, right? And the other point what I have observed, that if you have a cross functional leadership skills, then you might have good chances to rise in your career, right? So skill stacking, building a unique advantage. So what is skill stacking? So it combines technical skills along with soft skills. And if these skills will be combined, you will be stand out in your career, whether you are in a research career or in industrial career or uh, in managerial career, whether it would be any career. You need the amalgamation of these skills, combine technical skills with soft skills. So what is uh, what is a reason behind this concept, that it makes you stand out? So being technically strong is not enough in today's competitive world, right? To lead and influence and innovate uh, with a powerful mind, you need a powerful mix of technical and soft skills like communication, collaboration, as well as adaptability. Miss Snigdha has already talked about the soft skills. These are the major skills you need to stay and leverage a long term career in biotech industry or in any industry, right? And by combining these skills, you can develop various hybrid skills, like you will be more versatile if you combine your technical skills, like you are a data analyst, and your communication as well as collaboration and adaptability in new group, in new managerial roles, or in new leadership roles, then it will make you more versatile. It These things can make you more promotable. You can achieve more success, as well as more resilient in fast changing job market, right? So high value skills in 2025 are cloud computing, right? So the first skill is cloud computing, the new electricity, right? So cloud computing powers modern infrastructure, right? From biotech to artificial intelligence, to each and every field out around us is applicable in each domain these days, right? So importance of cloud computing is, it enables the scalable data analysis, computing, remote collaborations, as well as faster deployment of new technologies, right? If you are using chat gpt in your phones, right? And you are using a Gemini in your computers or in phones, right? Copilot in your laptops, then it all run with the cloud computing. And if you have cloud computing skills, you understand, you can develop as well as you can implement these skills, then you are fit for the current job roles, right? So this is the first skill of 2024-2025. Next comes in our field, that is the bioinformatics tools, the AlphaFold, Rosetta Fold, right? So these tools are the modern fuel for the biotech and pharma industries, right? These tools can predict protein structures, as well as these tools can model molecular interactions, right. And these tools are critical for drug discovery, precision medicine, and synthetic biology. So if you understand these tools, if you have skills, how to handle proteins, how to understand molecular interactions, and you have the basic knowledge of bioinformatics as well as cheminformatics, then these skills can be merged together, and you will be the best fit for drug discovery domain, whether these are industries or research labs or precision medicine running industry, right? And then comes the multi-omic analysis. If you have multi-omics analysis skills, so what is multi-omics analysis? It combines data from various biological layers. These biological layers could be genomics, proteomics, transcriptomics, metabolomics, right? So you have data sets from various domains, right? Here in genomics, you will have the information about genomes, that is DNA or RNA, right? Then in proteomics you have the knowledge or the data sets from protein structures, protein sequences, their folding, right? Their folding patterns and their denaturation patterns, their functional parts, right? So you must have these things in your mind. And if you are talking about transcriptomics, so the translational data, how a specific gene product is translating into the protein product, right? So if you have the basic knowledge of biology, molecular biology, cell biology, genetics, then you can attain these skills, multi-omics analysis skills, right? And then you can implement these skills into healthcare revolution and the healthcare industry, right? In these days you might be seeing various advertisements regarding PhD, regarding postdocs, or in industrial level, that the people are seeking the students or the employees with multiomics analysis skills, right? So these are the major skills in our biotech industry, or you can say the bioinformatics or cheminformatics or drug discovery, right? So my advice is, just don't just learn, apply. If you have learning these skills, right? If you for example, in these days youtube is the major resource of learning. People are getting learning new things, right? But they do not apply. You must have some projects, you must have some targets to achieve, to you must have some pipeline to develop, right? So after learning these skills, just apply them, build your portfolio project, right? Like Miss Snigdha has already discussed that, just go to GitHub, go to LinkedIn, and just you have, if you have developed your codes, you have developed your smaller pipeline for data analysis, for importing data, for cleaning data, for pre-processing your data, right? Then just upload it to GitHub, GitLab, or other platforms, right? She has already discussed. So it will let you develop your own portfolio for projects, that you can retrieve specific project, then you can handle it, then you can work on that project independently, right? So after having these skills, just apply and develop your own profile. So if you have a career in any domain of domain in any sector of industry, like biotech or any sector, then after having a beginning career, you have mid career crisis. Sometimes you feel stuck, you feel avoided, or you feel ignorant. Then at that time you have to go for lateral moves, which is the secret hack for your career. So what is lateral moves? So the lateral moves are shifting to a different but related role, same level but with new skills, where you will apply your skills to learn new skills, and it will it will enhance your horizon of skills, right? So a lateral move means shifting to a different but related role at a similar level in a new function or a new department with a new specific skill set of area, right? You are not moving up here, but you are moving strategically, right? With a specific strategy to attain a specific achievement later in your career, right? So why it works? It expands your skill set. I already told you that it enhances the horizon of your skill set, right? It expands your skill set without starting from scratch, right? So you are just going to different department, right? You are not moving to a new position or a higher position, right? You are just switching your department to learn new things. If for example, if you have if you have knowledge of chemistry, right, and you are working as a PhD and you want to learn about uh, how to work with chemical structures in computer, right? So you are starting you start learning about computational chemistry or structural representation or chem informatics tools, the basic chem informatics tools, right? So you will stay a PhD scholar, but you have a new skill set in your portfolio, right? Then it makes you a versatile candidate for future leadership. If you have chemistry skills, now you have computational chemistry skills plus you have what say chem informatics skills, then you will land a better job or better position in your future roles, right? So the lateral moves makes you more adaptable, right? They makes you more cross functional and make you better prepared for complex roles, right? So your next role, sorry, your next role doesn't have to be up, it can be sideways. So this is the motto, this should be a motto of your career while you are in your mid career journey, right? Do not always look up for the higher position or a senior position. Just try to work sideways and attain a new skills, then enhance your portfolio, and automatically you will land in a better and suitable role in your next career move, right? Or in your promotional roles. So when should you make lateral moves, right? That is the main question. So when uh, what would be the signs that now it is a correct time to make a lateral move, right? So these are the following three signs. So the first one is to broaden your skills, right? So when you have mastered your current role, like I have already exemplified that if you have chemistry skills, you know chemistry very well, you have organic chemistry things in your mind, you have inorganic chemistry things in your mind, as well as you have fundamental, okay, organic, inorganic, and you have fundamental chemistry, uh, clarity of these things, right? And now you want to expand your horizon to computational chemistry, chem informatics, and you can further implement these things into drug discovery, right? These are the applied roles. So the first sign is to broaden your skills, right? Here is I have just taken another example. If you are a data scientist moving to a product management to learn business strategy, you have already skills for data science. You can understand data, you can solve data, you can uh, make extract informational pieces from data, and now you want to learn about how to manage a product. Here you are a part of product development. Now you want to learn about that how business strategies can be made for product management, right? So this is a this will enhances your skill, and it broadens the application applicability rules. Okay, so the second sign would be to avoid stagnation, right? If your current role is becoming too repetitive or it lacks growth, right? For example, a cloud engineer stuck in repetitive tasks, right? So if for example, a cloud engineer, what a cloud engineer would do, they will maintain the cloud services, they will maintain the systems, they will maintain the servers or the backend programs, like, and then he can move to a particular cloud engineer can move to solution architecture for customer facing challenges, right? Then the third one is to unlock promotions. When your career growth requires more diverse and interdisciplinary backgrounds, like, so here I have taken an example that an bioinformatician transition to a computational chemistry. Now I was talking about chemistry guy. Now if you have chemistry roles, if you have bioinformatics roles, then you can easily transit into computational chemistry roles. Right, here what happens is, if you know about bioinformatics approaches, now forget chemistry guy, if you have bioinformatics background, right? So you must be aware of databases, you must be aware of programming, you must be aware of system engineering, right? Now you can easily move into computational chemistry tech domain, right? You have already knowledge of computers along with the biology. You can easily understand chemistry, you, because you already have foundations. If you are a bioinformatician, then you have already read chemistry in your past, at your graduation level or at your undergraduation level, right? Then just enhance your skills in computational chemistry, then it will let you gain cross disciplinary credit for a lead role, right? So the target adjacent roles would be uh, data scientist, right? So I was talking about the lateral moves. So here are the main examples for lateral moves. So what would be your adjacent roles? So data scientist, there I have already talked about product manager, right? So what a data scientist bring to a product manager role? So it will bring a strong foundation in analytics, data modeling, and user insights. Okay, so what that particular data scientist will learn in its new role, it learns the business strategy, market positioning, as well as product life cycle management, as I already told you. So how it will enhance your skills? So it will let you learn about a product, a role in a product leader, which makes a data driven decision at a rare and valuable combo. Okay, then a bioinformatician, I have already talked about the bioinformatics role, right? So in as a bioinformatician, what will you bring? So you will bring experience in genomics, sequence analysis, biological databases, uh, computer programming, software automations, software development, version control, etc. Right? Then what you will learn as a computational chemist, you will get to learn about molecular modeling, drug interactions, structure based drug designing, the protein structures, the protein ligand interactions, how molecules behave within a biological system by the simulation studies. So how it will enhance your skill set? So it will let you evolve into a cross disciplinary expert, which will be a key for innovation in biotech as well as pharma industry, right? So, pro, uh, I would like to give you a pro tip. So, study job description for target roles to identify skill gaps. So, whenever you want to land in a specific job role, okay. So, just try to dig deep into the advertisements, and try to find out that which are the skills you need in your career, and then that is the skill gap in your career, and try to learn those. Okay, so whenever you are on a table with us HR, where you are uh, going to join a new job, and you have to showcase your skill set and ask for a higher pay, then you can quantify your impact. So, how badly, what would be your bad saying, or how you can propose yourself as in bad manner? I have improved a pipeline. Don't say that. Just say that I have optimized a pipeline which reduced the cost by 30%, and I have saved 250,000 dollars per year. Okay, and then document your impact. I have built an automation and saved 15 hours per week. So this is just a simple example that how you have to make a negotiation. Okay. So pro tips in your career journey and at your mid career, learn the business, not just tech. Diverse your skills. That is a simple thing. Then find a mentor, two steps ahead. Always select your senior or your advisor or your supervisor, which who has a more experience than you, and who can let you think, who can push you to think in a new way and let you learn new things as well. Okay. Job descriptions are cheat codes. As I told you in a pro tip, that just uh, try to read in depth for a job description, where you can find out skill gaps and you can learn those skills to be a good fit in a current job scenario, right? And consider adjacent fields. So always go for sideways rules to enhance your horizon of a skill set. And the pro moves could be new team. Try to adapt or try to join a new team where you can get to learn new things as well as new skill set as well as visibility, right? Try to attend conferences, workshops, webinars, where you can connect as well as get to learn. Miss Snigdha has already discussed in depth there. And growth, these things are discussed by Miss Snigdha in depth. So with this thing, I want to thank you for listening me with the patience. Now I want to
Yes. Thank you so much, Dr. Neeraj. Thank
thank you,
you
thank you,
for that
thank you.
eye opening tour of all the skills and the moves, especially the lateral move. I'm sure many people out here, many attendees will find it useful. And also, we want to see now how these ideas are translating into large scale projects. So, I would like to welcome our next speaker, Dr. Girisha Verma, ma'am. Over to you, ma'am.
Thank you. Uh, Good evening, everyone. I'm Dr. Girisha Verma, currently working as a senior business analyst and product owner at Daiichi Sankyo. Um, Actually, I have uploaded my. Yeah, okay. Thanks a lot. Uh, Today I will focus on a theme that is becoming increasingly important as we scale scientific research and apply it to real world needs. Ms. Snigdha and Dr. Neeraj have focused on how to grab the job and then how to climb up the ladder, the career ladder. What I will be focusing on is leading bioinformatics at scale, which is more than data processing or pipeline execution. It is about aligning scientific strategy, applying systems thinking, and ensuring that all our bioinformatics work translates into clinical and business impact. Let's move on to the first slide, that is why leadership matters in bioinformatics. Let us talk about it. In the past, bioinformatics was often seen as a supporting function, someone who would just run the RNA sequence or process some imaging data. But today, as data becomes the driver of decisions, bioinformatics has moved into the strategic core of life sciences R&D. To lead in this space, we need to balance five key pillars. One of them is scientific strategy. As a leader, our first role is scientific direction. That means defining the right biological question. It's not about running every tool, it's about knowing what signal you're chasing and making sure the pipeline is built to uncover that signal. Second of it is technical integration, that is, tools, workflows, and pipelines. Leadership also means technical vision. Are your workflows reproducible? Are your pipelines optimized for scalability? Say in AWS or GCP? Are you using tools that match the complexity of your questions, like multimodal integration, machine learning, or spatial omics? A good leader does not just approve tool, they architect workflows that are efficient, compliant, and purpose driven. Cross functional collaboration is the third point. No bioinformatics project succeeds in isolation. We have to work with wet lab scientists to understand the experimental design, with statisticians for model validation, and with clinical scientists to define endpoints and patient stratification logic. So, leadership here means being the translator between disciplines. We speak data, but you also speak biology and business. Next, we have is regulatory alignment. We're increasingly working with sensitive data sets, clinical trial samples, patient derived xenografts, imaging repositories. That means we have to adhere many standards for HIPAA, GXP, and 21 CFR Part 11. A bioinformatics leader must ensure traceability, audit readiness, and data reproducibility. And that means building systems, not just analysis. The last point is soft skills. As already been discussed, we should not underestimate the people side. As teams get more hybrid, global, and interdisciplinary, we must be able to communicate across functions, mentor junior analysts, and resolve conflicts when priorities differ. The best technical teams fail if their collaboration structure is broken. Soft skills are not optional, they are force multipliers. So, why does leadership matter in bioinformatics? Because without it, the data stays data. But with it, the data becomes decision, pipeline becomes impact, and teams become movements. Coming on to the next slide, we have what it really means to lead bioinformatics at a senior level as a lead scientist or a director or a VP. These roles are not just about data processing or pipeline development. They require a strategic mindset, technical oversight, and systems level view of science and impact. First, we have a strategic scientific oversight at the leadership level. When we are responsible for designing the long term roadmap, what are the biological questions that my company or any company or research division needs to answer over the next 3 to 5 years? Are we validating a biomarker for early detection? Are we building a companion diagnostic? Are we designing AI models to stratify responders in the trials? You can also define success metrics. It's not enough to say model accuracy. Is your output reproducible across the cohorts, validated in wet lab or animal models, or is it IP protectable? Then we have pipeline and infrastructure management. Senior bioinformaticians architect workflows that scale. We are not just running RNA sequences on a desktop. You are setting up cloud native systems on AWS or GCP, as already been discussed, building Dockerized pipelines, integrating multi-omics, genomics, proteomics, transcriptomics, even imaging data. Currently, I've been working on the imaging data. These workflows must be scalable, secure, collaborative. Think of it as building the digital infrastructure of science. Data governance and compliance is third point at scale. Compliance is not optional, it is foundational. You must ensure that your pipelines are audit ready for regulators like FDA or EMA. That will mean using version controlled tools, documenting all the metadata, lineage, provenance, keeping logs of changes and access, who did what, when, how. Your infrastructure must follow frameworks, and you must train your teams to think this way. It should not be a burden, but as a safety and integrity principle. It should be founded whenever you start thinking strategically. The fourth point is cross functional collaboration. You're not leading code, you are leading people. So you must engage with clinicians. To define endpoints and clinical questions, we have to deal with wet lab scientists, statisticians, and regulatory teams. You are the translator between the disciplines. You take a biological need, like identify neoantigens, and convert it into the pipeline architecture. I'm sorry. You can also lead code reviews, data review meetings, and resolve technical bottlenecks before they escalate. For a real world example, as I have mentioned in this slide, let us say we are overseeing a neoantigen discovery project. You will start with FFPE clinical samples. You run mutation calling, you perform an HLA typing, and use ML to score immunogenicity. What would be the end output? Personalized vaccine targets for each patient. But as a senior leader, what our role is, it is to align the biology with the computation, oversee the pipeline design and validation, ensure that it is compliance activated, coordinate with the clinical teams to incorporate the results into the trial arms. In short, senior bioinformatics roles are not just technical, they are architectural. The design, scale, steer the scientific enterprise from question to clinic. Moving on to the next slide, we have defined the problem and choosing the right data modality. The most sophisticated algorithms or pipelines can't rescue a poorly framed biological question. As already I've said earlier, the first step is to define a problem properly. That's why the first responsibility of a bioinformatics leader, whether they are a scientist, director, or VP, is to formulate precise hypothesis driven questions. For example, which transcriptomic alterations underlie resistance to some drug in triple negative breast cancer? This is not just a data question. It reflects a clear therapeutic context, a testable hypothesis, and most importantly, it points out towards the data modality. Maybe an RNA sequence for gene expression or scRNA sequence if we are considering cell type specificity in resistance mechanism. There is a direct relationship between the scientific question and the tool we choose. A mistake in this early stage can cascade down into the wasted resources, inconclusive insights, or even misleading clinical correlations. This intersection of science and logistics, of hypothesis and sample source, is often where junior scientists stumble. And this is precisely where leadership makes all the difference. Let us now look at a real world controversy that highlights the consequences of vague problem definition. I'm sorry again. This slide shows one of the most famous and most misunderstood scientific controversies in the genomics, the ENCODE project. In 2012, the ENCODE consortium made headlines by claiming that 80% of the human genome has biochemical function. Only 80%. That was an explosive statement. You cannot just outright up and say that 20% of the human genome is of no use. It directly challenged the idea that large portion of our genome were non-functional, so-called junk DNA hypothesis. At first glance, this seemed exciting. How could people say that? We can discuss on that, we can have debates. But the backlash from the scientific community was intense. Why? Because ENCODE had defined the functional, very loosely. Basically, any DNA region that was bound to a protein or was transcribed got counted. Critics argued that this biochemical activity was not the same as biological meaning. Not every binding event or DNA transcript has physiological relevance. So, what is the takeaway for us? From this controversy, we can see as bioinformatics leaders, we must make sure our teams are not just generating the data, but asking sharp, refined questions. We need to know the difference between what is measurable and what is meaningful. This slide also helps students understand the risk of making headlines is not always a good thing if the foundations are not solid. If you are saying on the mic that 80% of the genome is functional and the rest of it is non-functional, you should have solid proof of that. As we move forward from selecting the right data modality, the next non-negotiable pillar is ensuring scientific rigor and validation. Let's move on to the next slide. In bioinformatics, especially in clinical or translational settings, our work doesn't end with generating results, it only begins there. So, first, we have scientific rigor. This starts with good experimental design. We are not just analyzing numbers, we are working with biological systems that are complex and noisy. To handle this, we use biological replicates, technical replicates, batch correction controls. Without rigor, we ask finding false positives or missing biologically relevant patterns altogether. And no matter how sophisticated our pipeline is, if the input is flawed, the output is meaningless. Next, we have reproducibility. We must use validated tools. Many examples have been given by Dr. Neeraj and Snigdha. Cell Ranger, Surat, Star, Descu, and so on. And more importantly, our workflows must be containerized and version controlled. That is, we should be always be using the updated version of any of the tools that we are using, like Docker, Singularity, workflows managers like Nextflow or SnakeMake, to ensure that anyone, whether in another lab or regulatory review panel, they can reproduce the exact results with the same data and code environment. Again, reproducibility is no longer optional. It is compliance requirement in many pharma setups. I have been working in a pharma company for so long, so I know this. Validation pipeline. Once we trust our computational results, we must validate them biologically. This validation is a multi-step, multimodal loop: in silico, in vitro, and in vivo. This layered validation makes sure we are not just publishing pretty heat maps, but generating real and actionable biology. Moving on to the next slide, we can see that connecting bioinformatics to ROI, let's now shift from strategy and validation to something equally important, that is what impact is going to take. In bioinformatics leadership, we don't just produce insights, we need to show how those insights contribute to ROI. Let me break it down further. By reducing target validation time with tools that reduce clinical dropout rates, and if you can improve cost per validated hit. As a leader, it is our job to frame bioinformatics results in terms of business value. As you can see in this slide, we are saving anywhere from $5 million to $10 million in development costs downstream. We can avoid expensive late stage failures due to poor molecular stratification, meaning the number of targets that actually go from preclinical to trial, your entire pipeline becomes more efficient. Moving on to the next slide, we have protecting innovation, IP in bioinformatics. As we scale scientific strategy, innovation is inevitable, but innovation is only as impactful as its ability to be protected. What is bioinformatics IP? Because in general, when we talk about IP, we talk about something new, something innovative that we are doing. But in bioinformatics, intellectual property refers to innovations that have either commercial value or scientific exclusivity. These are not limited to physical inventions. They also include the code, the pipelines, the curated data sets, and the models. We can have novel algorithms, thinking of custom algorithms tailored to normalize FFPE data under various scanner conditions. They should be documented thoroughly, versioned, and ideally backed by model training metadata to establish originality and reproducibility. If the algorithm can outperform standard tools or solve specific bottlenecks, it may be patentable or at least protected via trade secrecy. Then we have curated data sets. Let's say our lab has built a rare mutation atlas of pediatric cancers over 8 years. That data set is not just valuable, it is strategic IP. It's not just about the raw data, but the structure, annotations, and context applied to it. Controlled access, versioning, and access logs ensure both scientific credibility and IP protection. Then we have predictive models. Models built to predict drug-induced liver injury. For example, we have huge translational potential. If a machine learning model reaches high predictive power across patient cohorts, it could be protected as proprietary software or patented if its methodology is novel. At the very least, the model weighs training architecture and training data should be secured and version controlled. Coming on to our next story, we have the DeepMind AlphaFold licensing debate. Uh, as many of you know, AlphaFold 2, it was developed by DeepMind, a subsidiary of Google, made headlines by solving one of the biology's biggest challenges. That is, it predicted 3D protein structures from amino acid sequences with high accuracy. When DeepMind released AlphaFold's predicted structures covering nearly every known human protein, the community was thrilled. It was a breakthrough, obviously, but there was a catch. While the predicted structures were freely shared with the public, the algorithm itself, including its complete code base and training data, was not fully open sourced at first. We can have a debate on that because it was developed by salary earning people working in a company, but it was openly accessible to everyone. So there was a debate in scientific community whether private companies owned foundational scientific tools while public researchers only get the outputs. So after significant community pressure, the DeepMind did release the model weights and inference code, but the licensing remained restrictive, especially for commercial or derivative use. So here we have lesson for bioinformatics leaders. This case reminds us that bioinformatics algorithm ownership can clash with FAIR principles. Are we balancing innovations with openness? Are we enabling reproducibility? How do we license tools so that they can empower global science and not gatekeep it? For this slide, we can see the ensuring integrity, governance, and fair compliance. We see here, first point is data governance. The data governance ensures that data is traceable, secure, and audit ready. It's very important. Every transformation from raw fast queue to summarized result must be tracked. This is data lineage. It should be tracked. Tools like Git, DBC, Airflow help maintain this integrity. Second point we have is storage access control. It's not enough to store your data. You must control who accesses what, when, and how. We can use role-based access controls, implement cloud encryption, control vocabularies, and access auditing. ELN and metadata management can be done. For ELN and metadata management, like benchling or lab archives, are not just note-taking tools, they are regulatory records. Then we have compliance frameworks, fair principle reusability at scale. Here we can see that governance is not red tape. It is your insurance policy for scientific credibility, regulatory approval, and inter-team collaboration. For FAIR principle, as already I have said, so I'm skipping it. You know that FAIR is um, short form for findable, accessible, interoperable, and reusable. It's not just good science, it's a requirement for most consortia today. Coming on to the next slide. I'm sorry. We have system thinking, leading cross-functional teams in modern bioinformatics leadership. One of the most underrated yet critical skills is systems thinking. The ability to connect disciplines and orchestrate collaboration between very different stakeholders. Think of bioinformatics as the central nervous system in a biotech company connecting various limbs. There are many stakeholders. One of them is wet lab scientists. Then we have engineering department, we have statisticians, we have clinical teams. For wet lab scientists, we can ensure the samples we analyzed are of high quality. That will mean checking the RNA integrity numbers, validating sample prep protocols, confirming barcoding strategies before sequencing even begins. Then we have cloud and DevOps engineer. They are a backbone for the scale. We work with them to manage AWS or GCP compute environments to make sure our pipelines run efficiently. Then we have biostatisticians. We partner closely to validate models and ensure our analysis account for batch effects, confounders, and that statistical power is sufficient. Then we have clinical scientists. In translational research, we align with clinical teams to define patient eligibility, endpoints, and stratification logic. We have coordination tools like Jira, Confluence for planning, Nextflow, WDL. Finally, leadership is here about translation. You can translate biological hypotheses into structured data over workflows and translate complex results into decisions clinicians and scientists act on. Next, we have ethical imperatives in data driven bioscience. So, this is one of the most controversy prone topic. as bioinformatics become increasingly driven by AI and deep learning, we must address a critical area this critical area. One of the biggest concerns is model bias when training data lacks diversity. For example, it's an example when polygenic risk scores are built only on European ancestry. Models tends to underperform or misclassify individuals from other ethnic groups. And this is not just a technical flaw, it's a health equity issue. It cannot be the data that we have from European ancestry cannot be used for any other groups. The second risk is the use of black box algorithm, complex models where we can't explain how decisions are made. Then we have ethical pillars for responsible AI. We should have explainability. We can use the interpretable models or add explainability layers like SHAP or LIME to visualize feature impact. This ensures decisions are understandable to humans and not just the machines. We should have transparency. We should have informed consent and the data use. AI models trained on biobank or hospital data should stay within the boundaries of the original consent. Finally, from pipeline architect to talent cultivator, mentoring the next generation, let me close this leadership arc with what I believe is the most lasting legacy of any senior bioinformatician, not the pipelines we build, but the people we mentor. What true leadership means is developing others who can think critically, question assumptions, and carry forward a culture of rigor, reproducibility, and responsible innovation. We are not just teaching tools, we are teaching scientific habits. For core mentorship topics, we can have clean code and version control. We instill the habits like GitHub discipline, meaningful committed messages, modular code structures, and annotated scripts that others can follow and build on. Critical reading of papers, mentees should learn to spot statistical flaws, underpowered studies, and over-interpreted conclusions. We should have reproducible project environments using Docker, Singularity. We should encourage the next generation to contribute to open source projects, present at global forums, publish both code and data, join the communities where they can learn best practices and find the collaborators. Finally, to close this session, I want to leave you with one simple truth that is, in today's data driven world, bioinformatics leadership is not optional. It is foundational to progress in biotechnology. A true leader in this space just does not execute pipelines or interpret heat maps. They connect the dots between molecules, data, and impact. For scientific rigor, asking the right questions, designing hypothesis driven experiments, ensuring reproducibility across every step. We have technical depth from cloud native workflows to AI ML models. We have translational awareness. Leaders know how to turn complex outputs into actionable insights. And finally, whether you are pipetting at a bench or scripting in a cloud VM, your leadership is defined not by your title, but how well you orchestrate innovation that changes lives. Thank you for your time, and I hope today's session has helped you to not just see where bioinformatics is going, but how you can lead it forward.
Wow, what a power packed hour and what a tremendous session, Dr. Verma ma'am. Thank you so much,
Thank you,
Girisha
Vala.
ma'am, for giving us such a clear picture of what it takes to lead large scale bioinformatics projects and teams. Your insights into real world industry workflows, team management, and strategic thinking was truly valuable for everyone who are looking forward to grow into leadership roles. A big thank you to
Thank you.
you, ma'am. Yeah.
Thank you.
And I also want to thank our other speakers as well, Ms. Snigdha and Dr. Neeraj Kumar, for sharing the clear real-world advice on everything from scoring from your internship to steering multi-billion dollars biodata projects. So, a quick
Thank you so
recap.
much.
Yeah. So start strong, build a project portfolio and network early, scale up your AI, multi-omics, and cloud tools and must-haves, and think impact link every analysis to real business or clinical value. So, if tonight's session lit a spark and got you excited about real world bioinformatics, then you should join our bioinformatics internship, which is starting 30th June. This will turn your spark into proper knowledge to be there on your CV. You will work on live data sets, you will get mentor feedbacks, and you'll finish with a project which you can showcase in your job interviews as well. So, this is your chance to get hands on training in global research tools and techniques under the guidance of top experts. You can also choose from 30-day training or 3, 6, 12 months of project work. You can publish your research and gain real world experience with mentorship. So, whether you're aiming for industry jobs or higher studies, this internship will boost your skills and your resume as well. So, if you want to register if interested, please call up the numbers which are given here. Okay. So now it's time to open the floor for your questions. I'm sorry, but I won't be able to take many questions as we have already exceeded the time. So I have a few questions. Let me just scroll up. I think I have some questions before. Yeah, so this is a question from Maulik Chaudhary. How is pharmacoinformatics in Niger? Dr. Neeraj Kumar, will you be able to take up this question?
Yeah, sure, ma'am. Thank you. Thank you for asking this question, Maulik Chaudhary. So there are three NIPERs which take pharmacoinformatics students in MS level. So you have to first crack the GATE or GPAT, and then you can go for NIPER entrance test. And if you qualified, and you have to attend the counseling. And there are three NIPERs: NIPER Mohali, NIPER Calcutta, as well as NIPER Hajipur. They take the pharmacoinformatics admissions. And yes, you get to learn about various subjects like pharmacology, then the toxicology, then the programming in pharmacy, and how you can integrate your knowledge of pharmacy with the informatics to create and solve real world problems. So yeah, NIPER is a best place to learn about pharmacoinformatics.
Okay, thank you, sir, for your insight.
And
The next question would be from Nitin Sharma. Ma'am, how to land a PhD in bioinformatics right after BTech in biotechnology? Ms. Snigdha, ma'am, would you be able to take up this question?
Yes, sure, ma'am. Hi, Nitin. So after BTech, there are a lot of PhD programs that that are coming in, and that they are really interested in picking up just after the BTech. I think few of the exams that you should focus as GATE, CSIR, NET, IIT JAM, and all of that. Now, also, I think you should be, having a good statement of purpose. Also, a good, LORs and research proposals, good CVs for you. You can look up for, good institutes, and then showcase them all your SOPs and LORs, what you did. Yeah, you look for, who's the supervisor. The supervisors has which what kind of projects, what you are interested in. So definitely, with the right kind of SOPs and your worksheets, and a good exam, qualified, you can definitely approach the supervisors and the institutes. That is happening. They want to pick up students after BTech. So I hope this answered your question, Nitin.
Thank you so much, Snigdha, ma'am. Okay, the next question is from Sylvester's Oshin. I've completed B Farm and I have had a chance to study MSc in bioinformatics. Just learned something to do with pharma informatics. So, am I taking the right trajectory? Dr. Girisha, ma'am, will you be able to throw some insight on this question?
Uh, you have completed the B Pharma and you are currently doing MSc in bioinformatics. I think you cannot be on more right path. You are doing absolutely fine. But I would suggest you start with the soft skills along with your MSc. The few of the soft skills in bioinformatics will be given to you in the syllabus, I believe, but I think you should take up side projects as well. There are many platforms where you can take up the soft skills along with the projects, which will help you to increase your skills, but also might land you a good job in a biopharma company, bioinformatics field, especially.
Okay, thank you so much for that answer. Okay, the next question would be from just a minute. Yeah. Uh, yes. So Lakshmi asks a question: where can I do a part-time PhD in bioinformatics? So, Dr. Neeraj Kumar, would you like to take up this question?
Okay. So hi Lakshmi. So yes, you can go for part-time bioinformatics, PhD in bioinformatics. So there are few universities like I have known that MIT University, IGNU, as well as DY Patil University. These are the few universities which take up part-time PhD bioinformatics. But I would suggest you just go for full-time because you need to learn various things and you have to just focus on your research. So best of luck for your research.
Okay, thank you so much, sir. We have one more question. What are the best practices to build up a bioinformatics portfolio, and especially for a biotech undergrad who is going for MSc? Dr. Girisha, ma'am, any insights for this question, please?
Yes, of course. To build up a bioinformatics portfolio, you need to start with the softwares. I would suggest you start with Python. Uh, in well, informally we call it mother of all the computer languages. So if you understand Python well, you will understand all the other languages quite well. You can start with doing very small projects in R programming. That is also a very easy language to learn, and to go forward with with small projects. uh, Making projects in R programming, Python, and then moving on to the SQL. I think this would be enough to build up the bioinformatics portfolio.
Okay, ma'am. Thank you so much. And I would take up one last question probably. So I think this would be again for Dr. Girisha, ma'am. How can I enter into MNC using bioinformatics?
Oh well, you can always apply to the openings. But the thing is, you see, most of the companies that are MNCs, that are IT companies, mostly they will only use you. Basically, which are pharma driven, or there are some companies that are only linked to pharma companies, but are basically an IT company. So when you have a degree or soft skill for bioinformatics and you want to enter into MNC, you all you need to do is make up your portfolio, as Miss Snigdha has already I think focused basically on how to get the job in bioinformatics. Is that you can make your portfolio as good as with good projects that are already available. You can share my email ID, and I will guide you on many platforms that have the mentorship programs. They are free of cost, some of them, and some of them have projects that are related to specifically bioinformatics. They will lead you to the MNCs. When you finish the projects, you can always apply to the companies. Also, one more thing I want to add is, when you make your CV, when you make your resume to enter into the MNC, please use the latest keywords that are used, that the company wants, that are basically given in the job description. That is the main point. You have to use the keywords in your CV. I think this should be enough. Or you can share my email ID. I can put in more insights.
Thank you so much, ma'am. One last question. I think this has been posted multiple times. So this is the question from AI Pulse. How can I earn as much possible through bioinformatics? Snigdha, ma'am, any thoughts on this?
uh, Earning as much as possible. Oh, interesting question. So AI Pulse, I would recommend you to first, I think, choose a niche for you, a high demanding niche in bioinformatics. Choosing that helps will definitely help you. Say for example, genomics, AI ML for biology, deep learning, proteomics, clinical bioinformatics trending. I think you have to first select a particular niche for you, and then make notes out of it. What the industry wants. Drug discovery, AI ML. Maybe you can approach for PhDs, postdocs. To enhance your skills. Start with the career path. Bioinformatics. Uh, going towards the bioinformatics scientist thing, AIML, genomics, precision health, ed tech, and health tech is coming in. So maybe some freelancing consultant jobs are also there. So I think you have to select a particular path for yourself, niche of what you want to do. And then getting ahead step by step will help you, towards any of the niche that selects you select. So I think that answers your question.
Yeah, definitely. Thank you so much, Snigdha ma'am. So yes, BioBloom, that is correct, is the bioinformatics industry research internship. Yes, it has online hands-on training, as Dr. Neeraj emphasized, that you have to apply. It's not about only learning; you have to apply. So I don't think you will get a better platform than this. And thank you so much for your feedback and your words. So I'm glad that you found this webinar helpful. I think Srinidhi mentioned it, that really thanks to Biotecnika for this beautiful webinar about bioinformatics. I'm currently pursuing MSc Bioinformatics, so I'm confused about the future of career. So this webinar is really helpful. So with that, yes, we'll have to close the session right now. Once again, a big thank you to all the speakers. And final thought: remember, webinars like this are more than just sessions or lectures. They are your career checkpoints that save you months of trial and error. Okay, so Biotecnika lets you know the actual things what you are going to face in the industry, right? So keep learning, keep asking, and keep climbing the ladder. Thank you so much, everyone. With that, we come to an end. Good night, everyone.
Thank you, Urmila. Thank you, everyone.
Thank you, ma'am.
Thank you.